So I think we can get started and attendees will continue to trickle in. So hello, everyone. Good afternoon, good morning, good evening, whatever time it is for you. Welcome to this webinar with Boehringer Ingelheim and BioMed X Institute / XSeed Labs about our call for application on next generation clinically relevant models of fibrosis and wound healing. My name is Rita Freischlad. I am the BioMed X Events Manager, and I'm based at our institute in beautiful Heidelberg, Germany, which you can see behind me and Christian. On the panel today, we have Christian Tidona from BioMed X Heidelberg, Alessandra Barassa, John Broadwater, Gaurav Kaushik, Jill Steinbach-Rankins, and Yu-Chen Kim from Boehringer Ingelheim, as well as Mark Johnston from BioMed X USA. So Christian, maybe you would like to start us off with a quick round of introductions.
Yay! Welcome everyone. Thank you very much, Rita. Great having you at this webinar. As Rita said, my name is Christian Tidona. I'm the founder of the BioMed X Institute here in Heidelberg, and I'm happy to be with our colleagues here from Boehringer Ingelheim and Mark Johnston. Maybe over to you, Mark, as next.
Sure. So Mark Johnson, everybody. So my undergraduate background is engineering. I have a MBA from the London Business School, and I've spent the last 20 years running or starting 6 different businesses in the cell and gene therapy space, and I'm now running the US operation. I'm really happy to talk to you further about that.
Great. Alessandra?
Hi, everybody. Alessandra Barassa here. I'm the Global Head of Innovation Technology at Boehringer Ingelheim. You see the site in my background, and since I have a slide on my background, we'll talk about me a little bit later.
Thank you. Jill?
Yeah, so I'm Jill Steinbach-Rankins, and I'm a Senior Research Fellow in the Materials and Analytical Sciences Department at Boehringer Ingelheim.
Great.
Thank you.
John?
Yeah, hi, I'm John Broadwater. I lead an in vitro pharmacology group in our cardiometabolic diseases research department.
Thank you. Gaurav?
Hi, everyone. My name is Gaurav Kaushik. I am from non-clinical drug safety department from Boehringer Ingelheim Ridgefield. Hi.
Thank you. Yu-Chen?
Hello, everyone. My name is Yu-Chen. I'm a principal scientist at Material and Analytical Science Department.
Great. I think that was everyone. So before we start, some brief housekeeping. If you look at your toolbar in the Zoom window, you'll see a little Q&A box. We ask that you enter all of your questions using the Q&A box and not the chat. You can enter questions there throughout the talks. You can also like and upvote other people's questions that you find interesting. We will go through all of your questions when we get to the Q&A portion of the webinar, but as I said, you can type them in as you think of them. And next, you can find further information on the call, including some helpful links in the chat. So I'll post those in just a moment. And we are recording the webinar, and we'll send a link to all of you tomorrow and post it on our BioMed X YouTube channel very soon. And finally, after the webinar, if you have further questions on the application process, you can email my wonderful colleague and our communications and recruiting manager, Bianca Christian, at fbc@bio.mx, and I'll put her email as well in the chat. So with that, we can get started. Christian, over to you.
Thank you very much, Rita. Let me quickly try and share my screen. Here we go. Can you see it?
Yes.
Okay, sounds good. Good. Once again, also, as Rita said, we're recording this this webinar, and you can, if you have didn't understand all of it, you can always watch it again or mail us. Let me quickly try to move to the next slide. Here, before we start, a little information about myself. I always had 2 hearts in my chest, one of a scientist and one of an entrepreneur. Did my PhD in Heidelberg in the late 90s, but then during my PhD started my first company, which was a diagnostics company. And then joined the cell therapy company. This is where I met Mark, who is now our CEO for the US. I also started the Stem Cell Institute, Heidelberg Institute for Stem Cell Technology and Experimental Medicine with Andreas Trump from EPFL Lausanne. This is still active in Heidelberg. Also, I was for several years the head of the local cluster, managing an 80 million euro public-private research and development fund. And in 2013, I wanted to do something differently, something that creates impact, especially following one of my passions, which is helping research talents to grow. And this is what BioMed X is all about. It's about early career scientists who are curious, who want to develop themselves and who want to learn about alternative career paths in industry and also in startups. And this is what we built BioMed X Institute for. We started 2013 on the campus of the University of Heidelberg. Meanwhile, we have 2 international sites, one in Israel called XSeed Labs and one in the US in New Haven, Connecticut. And very soon also with this call, we will have a presence on the campus of Boehringer Ingelheim in Ridgefield, Connecticut. In Heidelberg, we're embedded in the campus of the university. This is very important for our model because we want to bridge the gap between academia and industry. So on one hand, combine the vibrant ecosystem of an academic campus or also of an industry campus together with the know-how how to turn great research into something that will ultimately help a patient. And we do this using a new innovation model, which consists of 2 major elements, global crowdsourcing and local incubation, which I'm going to explain a little later. And we published this new innovation model in Nature Biotechnology in 2015. Now, the way it works is when you go online, you're going to find a call for application. And this is how we usually start projects. We publish a very tough preclinical research question worldwide at the best universities and research institutions and invite early career academic scientists to apply by submitting a very original project proposal, how to solve this particular problem. And usually we get between 100 and 200 project proposals from 60, 70, sometimes 80 countries. So it's truly global. And then together with our pharma partners, we select the 15 brightest individuals and we fly them in for a 5-day boot camp to one of our locations. And then at the end of the boot camp, they pitch their presentation, their project proposal in front of a jury, which includes the senior management of the sponsoring pharmaceutical company. And the winner of this bootcamp then receives a major research grant and is moved with his or her family to our location. We organize visa, jobs for their spouses, kindergarten spots, new homes, whatever they need. And then they live and work with us for a time period of up to 5 years. This is an image of last week where we had our first bootcamp in the US at our new site in New Haven, Connecticut. In the back, you see part of the campus of Yale University, and you see that this is really an exciting experience with great scientists, but also with a lot of fun. This was a bootcamp as a topic sponsored by AbbVie, which was in a field also of tissue engineering, but in this case, it was very much focused around inflammation. This is the call for application, what this webinar is all about, which you will see on our career space and also online. It's around next generation clinically relevant models of fibrosis and wound healing. There's still 28 days to go. The final group will consist of a group leader, 2 postdocs, and 2 research assistants, so 5 people. And the winning group leader will get a budget of $890,000 per year for a time period of up to 5 years. What's very important for BioMed X at all of our locations are our core values. And this is also why we are performing those boot camps. We want people who are not just scientifically brilliant, but who have embraced the concept of growing by helping others. This is what cross-leverage means. We inspire and motivate each other to unleash our full potential. So BioMed X is a strictly non-toxic environment where we help each other and where we want you to grow as scientists and also as people, as leaders. Second is cross-pollinate. We embrace diversity as it is the source of innovation. For us, it's very important that people are looking upon our very tough challenges from all different perspectives. And this is not different scientific backgrounds just, but it's also different backgrounds where they come from, what they believe in, and what gender they have. So if you come and visit us in Heidelberg or in New Haven, you will see we're all a little different, all a little crazy, and it's good like that. We celebrate diversity. And also, and that you will also find out if you're invited to our boot camps, aim high. our goal is to enable a new generation of targeted therapies. We put a lot of effort together with our pharma partners to provide really all the resources you need for great science and to create something that could be a starting point for a new drug discovery program helping patients. And of course, we expect you to make the best possible use out of this time at one of our institutes or one of our sites. Little bit of statistics. Since we started, we've worked with 9 different pharmaceutical companies, and also we have our incubator in Israel. In Heidelberg, we have started in the last 10 years, 21 research groups, each on average with a €1 million, $1 million budget per year. So you can do the math. Pharmaceutical companies have invested roughly 100 million in the last 10 years into our research groups and into our programs. We've also started already 4 startup teams in Israel at Ion Labs. Ion Labs is a little different. I'm going to explain you later. It's for generating startups. We also publish. It's very important for us because all of our teams are performing very deep state-of-the-art science. And it's very important that we publish this science because at the end of a BioMed X term, you should be able to choose your next big step, either in academia or industry or startups. So everything should be open. And of course, not publishing is not very good if you plan for an academic career. And we're also educating. We have completed more than 60 bachelor, master, and PhD theses in the last 10 years, and all of our research teams have at any given time several master, bachelor, PhD students. We focus on deep disease biology because we are convinced that, as you might know, in this field, we're very good at curing mice, but not so good at curing humans. So most of our teams are focusing on deep human disease biology, and artificial intelligence is also very important at the moment, as you know. Our research focus is in 3 big areas. One of them is human organotypic disease models. This is also very important for this particular call, and you will hear that a little later from our partners at Boehringer Ingelheim. We have very wide access to primary human materials. Some of our teams get whole human organs from multi-organ donors, and they build organotypic disease models. On the left side here, you see the Mimeta system, but of course, there's many other organotypic culture models, even ones that are much more complex. All of these teams are working in deep disease biology, working with single cell in spatial and temporal resolution. And of course, these teams need also bioinformaticians and computational biologists, statisticians that make sense out of all of these data with cutting edge AI and machine learning tools. And the third part is sensor development. Some of our teams are developing electrical, optical sensors and so on. And you need to imagine that all these teams collaborate and help each other. And this means that this is a very fast innovation process because you get help and support from all kinds of disciplines. A couple of words about Ion Labs. There's a major difference with Ion Labs in Israel. If you win a boot camp in New Haven, Ridgefield, or in Heidelberg, you will become a full-time employee of our BioMed X Institute and get a research grant. If you win a boot camp in Israel, you become the first employee and shareholder of a new company in which the new IP is going to be developed. The focus in Israel is entirely on artificial intelligence for drug discovery and development. So these are all dry lab teams with a little smaller budget. Budget on average is $500,000 per year. And also a difference is that these projects have multiple sponsors per project, multiple pharma companies, and also Amazon is a partner in an Israel venture capital fund. And we have, for the next 5 years, we have enough funds to start 20 of those startups, and all of them will also be published in our career space, and you can apply. As we are Mark already mentioned, we have a new presence close to Yale University in New Haven, Connecticut. Our first boot camp, as I said, was really a blast. And we're intending to grow this quite rapidly. And of course, subject of the webinar today is our very new model with our partner, Boehringer Ingelheim. Boehringer Ingelheim has been a partner for a long time of BioMed X, and we have jointly decided to try something new together, which is putting the BioMed X model in a box on the campus of a major pharma company with all the local environment. And this is called XSeed Labs. And this is what the project of today is all about. So the winners of the first XSeed Labs call will be present on the campus of Boehringer Ingelheim in Ridgefield, Connecticut. And so why should you apply? These BioMed X projects are all fully funded 3 to 5 year research groups with quite generous budgets. They focus on outstanding research and publications, so no teaching obligations or grant writing. You are in a creative cross-cultural teamwork atmosphere. You get guidance by experienced mentors from academia and from industry. You're in an outstanding environment with direct access to leading research institutions and companies and also their core facilities and assets. You get leadership and entrepreneurship training, and of course, it's a risk-free assessment of a potential future career in a startup or a big company, because, as I said, we also publish, and you can also always go back to an academic career and become a professor. Can we attract top talents from around the world? I think 10 years down the road, and after 300 alumni, mostly in Heidelberg, I can say, yes, we can. These are just some of the logos where people came from before they joined BioMed X. And where do they go? About 50% go to industry and 50% go back to academia and become professors or top scientists. So with that, I would like to close. If you are interested in our calls and our job offers, all of them are published on our career space under career.bio.mx. And of course, also, I'm always happy to connect with participants of our webinars and bootcamps on LinkedIn. So please, if you like, send me a request. And of course, also you can follow our page on LinkedIn of our BioMed X Institute. And with that, back to you, Rita.
Thank you, Christian. So next up, we have the Boehringer Ingelheim speakers. So over to you, Alessandra.
Sounds good. Thank you, everybody. Can you see my screen here?
Almost.
Getting there. There we go. So thank you so much. Thank you, Christian, for that overview. And I'm very excited to be here. I'm going to take you through 1 minute of who I am, and most importantly, the company Boehringer Ingelheim. And then even more importantly, I will have my colleagues, my 4 colleagues, introduce themselves. They are going to be your industry mentors, and they are going to show you what challenge we're facing and how we would like to see these resolved. Just a minute about myself. I have a history in science that took me crisscrossing across the ocean a few times. It starts at the University of Padua, where I received a degree in organic chemistry with work around natural antibiotics that use structural changes to be more effective. With that work, I then went to Harvard Medical School, where I spent some time using that type of thinking to PTH, so parathyroid hormone, which is now a commercially, a fragment of which is a commercially available molecule for osteoporosis. I then came back to Europe and I did my PhD at the Max Planck Institute in Munich for biochemistry. And here I was working on inhibitors for BACE, for beta secretases and MMPs, so Alzheimer and cancer if you want. If you see throughout this work, I love science, I love cutting edge science, I love new technologies, but I also wanted them to be applicative directly to people and to patients in need. So I decided for my postdoc to take a translational work in a double position at NYU and Mount Sinai. And there I was working on human HDL that was loaded with imaging agents to image atherosclerotic plaques in mice and in humans also. After my postdoc, I jumped to industry where I thought science can really be applied, and I joined Bristol Myers Squibb initially, first in chemical development, and then I grew in various positions to global product development. And during my tenure at Bristol Myers Squibb, I also completed an MBA at Columbia University. A few years back, 3 years and something back, I saw a very interesting position at BI, and this is how I got to BI. And I am now responsible for innovation technology in the development space, which means that I lead a group of extremely talented scientists that are located throughout the planet. So we have people in Germany, in the US, in China, and in Japan. And they really look around everywhere to try and find new technologies that can help us develop our molecules, the molecules that are in our pipeline, and help also companies that maybe have ideas, but they're not fully developed to really make the next steps ahead. Let me move on from myself and move to Boehringer Ingelheim, which is a mouthful for people not speaking German, so you can call us BI, and that will be just sufficient. We are an interesting company, if you want, in the pharma space, because we are the largest private company. So we are family owned. We are still owned by the same company, Boehringer. We have a long history. We were founded in the late 1800s in Ingelheim, Germany. And our focus is, we have three major focus: human pharma, by far the largest. And then we have an animal health component, about 20%, both in revenues and in employees. And then a contract manufacturing organization for biopharmaceuticals. Family owned, I mentioned, and this is not just some interesting thing. It actually has a direct impact on how we do work. If you can imagine the spectrum between academia and industry, you can place BI kind of in between. And the reason is that we are private. We don't have pressure from Wall Street. We don't have to follow quarterly calls with investors. The investors are within the company, are the family. We are directly involved in their decision. And this allows us to really focus on what we need to do best, which is the science, the technology, the innovation, and really having an impact on patients. We are also not exactly a mom and pop type of business, even though we are family owned. We are over 53,000 employees worldwide. And then I put in this slide a few other numbers that might be of interest to you. More interesting is the impact that we have had on the planet. There are, of course, with such a long history, we have a number of products, and I'll drag your attention to the latest ones. So we are very active in type 2 diabetes, for example. So we have Jardiance and Trajenta. We have in COPD Spiriva. Another very important product for us is a blood thinner, which is Pradaxa. And then our latest molecule that we just got approved last year as the first molecule that can help patients affected by generalized pustular psoriasis, and that is Pervigo. And here you have a bunch of other products that might be of interest. We have 25 R&D centers, main R&D centers, I should say, and those are spread among 5 companies, so 5 countries, I mean, so the US, Germany, Switzerland, Austria, and Japan. So a pretty big R&D structure, if you want. As I mentioned, what we are really focusing on is innovation science, and this is the strong emphasis of the company. And the fact that we don't have to respond to investors and to Wall Street allows us to really think in generations. This is a buzzword that comes in the company a lot, because really, if there is a reason to continue with a project, if there is a reason to invest in certain science, as long as it has an impact on a generational scheme, if you want, to either patients, to employees, or to the planet, that will be received positively by the management of the company. We are particularly active in 6 therapeutic areas that you see listed here, but that doesn't mean that these are the only areas that we are focusing on. Again, focus is on science, focus is on technology, and the company invested on a group, RBB, the Research Beyond Borders group, that just goes around and independently of what our strategic decisions are, or independently of what our pipeline has to follow, if there is science or if there is technology that can benefit patients, we have the authorization to go ahead and pursue these new technologies. We are very excited to be here with BioMed X, as Christian mentioned earlier. this is a very new and innovative incubator model that we are subscribing to. You might look around online and you will see that there are incubators in industry. There are plenty of incubators in industry, but what normally happens is that you have a building over there, somewhere in campus, but over there, not with the scientists of the company. And they do the science, they do great science, but they are not really integrated. What we are trying to do here is a new way of really interfacing academia and industry together. And the reason is that we want to really foster this dynamic ecosystem that leverages synergies and out-of-the-box thinking to really access transformational ideas and solve complex problems. So as a BioMed X scientist, you will not be over there. You will be in a lab that is really next to BI scientist labs. So you will be really an integral part of the BI scientific community. And we are very excited. So there is no difference basically with our scientists. And this also implies that as a BioMed X scientist, you will have access to everything that the BI scientists have access to, including the core facilities that we have, including specialized labs, and including all the advanced equipment that the site that we have in Ridgefield offers, or even in Germany, as a matter of fact, or in Japan and in other areas. Here I put a list of some of the equipment, nowhere near exhaustive. This is just an example, but the bottom line is, and the take-home message is, as a BioMed X scientist, you will be an integral part of our scientific community. The next part here is really the most important part. So John, back to you on describing the project that we are excited to start with BioMed X.
Thank you, Alessandra. If you could go ahead to the next slide. We've, at the beginning of this webinar, we presented ourselves and our names, and you'll see from my colleagues here in the next few minutes just exactly what we're interested in. That's what we'd really like to get into. If you go to the next slide, really the simple answer is that what we're really interested in is trying to get a next generation model that captures the full aspects of fibrosis, fibrogenesis, fibrosis resolution, and wound healing. And then ideally, if we have such a model, we think it'd be fantastic if we could take a look at drug delivery as well. So there's some unique challenges that we face when trying to deliver therapies to fibrotic tissues. So if you go to the next slide, just briefly, just to set the stage, I mean, I work in cardiometabolic diseases where I have certain interest in certain tissues, but in speaking to my colleagues across therapeutic areas, we quickly realized that fibrosis is really a common driver of a lot of the pathologies that we're trying to treat, not only in Boehringer, but also in pharmaceutical industry in general. And secondly, there's a lot of processes that go on that we just don't really have the ability to model in vitro. And that makes it difficult to identify new concepts and to really understand what's going to be the next therapy that's going to help patients. Another element to this is that even if we come up with an antifibrotic that looks attractive, is we need to understand what the potential risk is for getting in the way of natural wound healing processes. Because obviously a lot of patients are going through lots of different challenges, for example, surgeries, and you want them to be able to respond to that with normal fibrotic wound healing. And this is another key area where we lack the ability to assess the risk of a particular antifibrotic. Is it going to interrupt with natural wound healing or not? So really what we're looking for in this ideal scenario is an in vitro model or models that would help us to identify and evaluate antifibrotic therapies and to assess their risk to impact wound healing. And as I mentioned at the start, there's also a lot of interest from our side to understanding how we might deliver therapies to fibrotic organs. And for that, I'm going to hand it over to my colleague, Jill, to speak about this.
Yeah, hi. Thank you so much, John, for that introduction. And so really here, in addition to a better mechanistic understanding of fibrosis and wound healing, multiple challenges really need to be overcome to translate this information to effective anti-fibrotic therapies. And one of the biggest barriers to successful treatment is that the microenvironmental complexity of fibrotic tissue makes it challenging to obtain therapeutic efficacy. And so namely, the genetic markers, biochemical cues related to fibrosis and wound healing result in irregular spatial and temporal differences in the tissue microstructure, as depicted somewhat on the right, that create hurdles to both obtaining and predicting therapeutic transport, efficacy, and ideal drug delivery strategies. Moreover, the multiple cell types involved in the stages of fibrosis make it difficult to target specific populations and stages of fibrotic signaling and progression. Hence, the mission of what we seek to obtain is an improved experimental understanding of and the ability to predict therapeutic transport within the heterogeneous fibrotic microenvironment. And the impact we envision from this is that these discoveries, together with available preclinical and clinical data, can bridge fundamental scientific findings such as the omics and biochemical pathway data with an application-oriented solution relevant to drug product performance that really helps us to inform and design the selection of APIs and formulations that maximize targeting to intended locations. And in turn, this would enable us to develop new APIs and treatment regimens that can overcome transport barriers and result in more efficacious patient treatment strategies. Next slide, please. So we'd like to leave this call open for proposals and leave it pretty broad to encourage innovative and transformative ideas. However, components of one potential approach may be to measure transport of relevant therapeutics through various stages of 3D fibrotic tissue models. Utilize these data then to develop a mathematical model that quantitatively predicts microenvironmental or other parameters that impact efficacious delivery. Integrate these experimental and modeling data findings with preclinical and clinical data, and then using these data sets, validate and refine modeling results to enable the rational design of formulations that localize delivery to intended locations. Next, please. So really here, what we were hoping to capture is from a drug delivery perspective is big picture. We envision that the proposed next generation model may incorporate an approach that enables measurement or quantification of therapeutic agents through 3D in vitro fibrotic wound healing models of various pathological stages, and that this will provide an enhanced understanding of limitations surrounding and strategies to improve delivery. And then we can see that in parallel, generating this ability to predict therapeutic transport and distribution within a mathematical model that considers things like maybe tissue heterogeneity, molecule properties, may help to inform the design and selection of APIs and potential delivery strategies to, again, maximize tissue targeting. Importantly, this will enable us to better understand and translate gaps seen in efficacy and toxicity, while also highlighting key attributes with a model that can be trained and validated to automate or to rapidly screen for different solutions. And then together, these discoveries with available, again, preclinical and clinical data, potentially across different therapeutic routes of administration, will bridge fundamental scientific findings to overcome these barriers and result in more efficacious patient treatment. So again, we thank you so much for your interest in our project, and we're really excited to see your innovative proposal submissions. Thank you.
Good. Rita, are you still there?
Yes, sorry.
Oh, no problem.
Okay, thank you very much to all of the BI speakers and to the attendees. Please feel free to start typing in your questions. So now you've learned a lot more about the challenge and about BI and about BioMed X. So hopefully some questions are starting to stir. And for the last talk, before we get to the Q&A section, I'll hand it over to Mark to tell us more about XSeed Labs and what to expect from BioMed X USA.
Great. Okay. Presentation. You see it?
Not yet, but it's loading. Still loading. Maybe exit and try it again. The share screen mode. Stop it and restart it.
Yeah, I just got a crash report. Hold on, let me
All right. Sorry, everyone, technical difficulties.
Oh, something's coming. Yep.
Here we go. All right, we can see it. Mark, we don't hear you. You're on mute and your camera is also off.
There we go.
There we go.
There we go. Okay, yeah, I got kicked out, so I had to sign back in. All right, so what I like to do is the first slide, I want to do my life, my work, my passion. This is an exercise that all BioMed X employees go through to give an overview of themselves. And for those of you that win a ticket to the boot camp, you will also be doing this exercise. So I thought I'd start there. So I'm very passionate about work. If I'm not excited about it, I won't do it. So I was really, really happy when Christian contacted me last year and asked me to set up the US operations, because what we're doing at BioMed X is so cool. And for those of you who haven't had a chance to go to the website, please go take a look at it and see all the really interesting science that we're doing. Family is the most important thing in my life. I think you'll find that out about me after working with me for like 5 minutes. I'm a big lover of travel. I've lived in Barcelona, London, Mexico City, New York, and I currently live in Los Angeles, but moving to Connecticut in September. So excited about that. I visited 6 of the 7 continents that we have on our planet. So travel's very big for me. And I'm really an exercise fanatic. So I love hiking and biking and really, really love hot boot camp classes. I live near the beach. So for me, when I was looking for places to set up the US operation, to be frank, Connecticut did not jump out at me because I hadn't been to Connecticut in over 20 years. And I think I was there for maybe 1 day. So that all changed when I visited. Let's see. So there's so much to do in Connecticut and Ridgefield area in particular. We have hiking and biking, golfing, skiing, tons of aquatic sports, and running. And that's all within, you know, a 15 to 45 minute range from the Ridgefield area. So there's just so much to do, and it's very, very appealing to somebody like myself who really loves the outdoors. So hopefully you'll find that appealing as well. There's plenty to do in Ridgefield. Beyond that, there's 70+ restaurants in Ridgefield and the surrounding area, from American to Greek to pizza. This is a very big thing in Connecticut. There's 30+ bars and clubs in the general area, and there's festivals all year round. And I think you will love it as much as I do. So the next question is, why work on BI's campus? Well, there's so many advantages that we think we're going to be able to take advantage of. And that includes daily access to the BI mentors. That, I think, is something that we don't currently have with the BioMed X teams. We have frequent interactions with our mentors at the other pharma partners, but in this new model with XSeed Labs, having that day-to-day interaction, I think, is only going to be beneficial. And as Alessandro said, you're going to be right next door to, your lab is going to be right next door to the people you're going to be working with. And already she showed you the access to state-of-the-art laboratory equipment and the intense core facilities that you'll get access to. I mean, these are huge advantages for you. So this ought to stimulate, success for the project. You'll have access to the scientific seminars that are on campus at BI. There's multiple cafeterias with, and this is really cool, subsidized food and beverage options. I mean, you're really getting a very, very good discount when you go to the cafeterias every day and interact with your coworkers. There's gym and health club access. There's free parking, and then daycare facility is available on campus. And then within the state of Connecticut, it's very strong bioscience workforce. That really allows you to source local and quality talent for your teams. We're a member now of BioConnecticut, and they strongly support R&D with discounted pricing on services. They have their own seminars and monthly working events. So this is going to help us to really plug into the overall ecosystem that exists not only in Southern Connecticut, but also within the entire state. And then there's just really high quality of life in the state. I mean, we're number 3 for K through 12 education, number 5 for quality of life in the US, 4th lowest crime rate in the US, one of the best places to raise a family. And as you can see, hundreds of miles of scenic coastline and thousands of miles of hiking and biking. So if you really like the outdoors, you're going to love Connecticut. And if you ever get bored and you want to get out of town, New York City is in some cases less than an hour away. You can drive or take the train, and there's tons of museums and theaters. And I'm sure everybody's, you know, at least watched a movie or television show where New York is described. So if you haven't been there, you'll love it there. There's international flights from Newark and JFK airports, and camping and beaches are only 30 minutes away. So I hope I was able to, show you the benefits of working and living in the state of Connecticut. Is really one of the greatest places to live, and you'll see that if you come work with us. Thank you.
Thank you very much, Mark. Very nice. So now we're getting to the Q&A portion of the webinar, and a few questions are already trickling in.